Hello everyone, welcome to another video. In this video, we will see Hershey and Chase experiment. Hershey and Chase were the scientists who were doing research on bacteriophages. Bacteriophage is the virus that attacks upon bacteria. Bacteriophages are composed of both DNA and protein, and they replicate inside the host body within 20 to 25 minutes. Scientists who were doing research on these bacteriophages wanted to know that which molecule of these bacteriophages, DNA or protein, is responsible for the replication, and which molecule, DNA or protein, enters inside the host body, or the whole bacteriophage enters inside the host body. So Alfred Hershey and Martha Chase set out an experiment for this purpose. They used radioactive elements phosphorus 32 and sulfur 35 to label the DNA and protein of bacteriophage. First, they labeled the protein of bacteriophage with sulfur 35 and allowed these bacteriophages to attack upon bacteria in this flask. After that, they did centrifugation. Centrifugation is a technique which is used to separate different components of a mixture. So here they separated bacteria from the bacteriophages. And what they found? They found bacteria in the pellet without any radioactive element sulfur 35 in them, and in supernatant they found bacteriophages. And bacteriophages have the sulfur 35 radioactive element in them. It means that sulfur 35 radioactive protein is not transferred from bacteriophages to the bacteria. So protein is not responsible for replication. Protein is not the hereditary material. It proved here. They labeled the DNA of bacteriophage with phosphorus 32 and allowed those bacteriophages to attack upon bacteria in the medium. And after they attacked, they did centrifugation. They separated out components of bacteria from the medium. And what they found? They got bacteria having phosphorus 32 labeled DNA inside of it, and bacteriophages in the medium having no radioactive element phosphorus 32. So they moved to the conclusion that DNA is responsible for the replication of these bacteriophages.